Hello students, today we are studying a topic for standard 10, biology, how do we see? Now for you, I have drawn a simple diagram here to understand how our eye functions. As you know, when we studied the structure of the eye, we saw the various parts of the eye. We also came to understand that the eye is an organ of sight. It's a sense organ which deals with sight. Now how does this eye perform its function of sight? Let us study this now. So here you can see, I have drawn here an object for you and a source of light. The light rays are falling on the object. For your simplicity of learning, we have drawn only two rays here. Actually, many rays are falling at various distances, so they are getting reflected and entering the eye. Now the first structure that meet these rays of light is the conjunctiva, the outermost covering. It is even covering the cornea. Hence, first the light rays fall on the conjunctiva. From the conjunctiva, the light rays enter the cornea. The cornea, as you know, you will have seen the structure of the cornea. It is a curved structure. It has a curvature. Because of this curvature, it helps to converge the rays of light. So the first structure that helps in converging the rays is the cornea. Now, the cornea, further from the cornea, the light rays enter the aqueous chamber. The aqueous chamber is filled with a fluid, a watery fluid called the aqueous humor. The aqueous humor, it also again helps in continuing with the convergence. So all these parts, they help to converge all these aqueous humor, cornea, all these help to converge the light rays as they enter the eye. Now after the aqueous chamber, which is filled with aqueous humor, the rays of the light via the pupil. Where is the pupil? It is located in the middle of the iris. It's an opening in the iris that allows light rays to enter and fall on the lens. So the light rays come through the pupil, they fall on the eye lens. The eye lens, you know, it's a crystalline structure. It is located after the aqueous chamber, and the pupils allow the light rays to fall on the lens. From the lens, the light rays travel via the vitreous chamber. Vitreous chamber is filled with a jelly-like substance. Again, it helps in convergence. It stops the scattering of light and it helps in converging. From the vitreous chamber, through the vitreous humor, the light falls on the retina. The retina is the light-sensitive pigment containing layer of the eye. What are the pigments contained here? The rods and the cones. These are light sensitive cells. They get stimulated by the light that is falling on the retina. Now it has come through this entire system. All this together from here to here, these are all parts of the eye. So what are these? Together they form an optic system. Now as we come to the retina, the rods and the cones in the retina are connected with nerve fibers. So chemical stimulation, the light stimulation is again changed into a chemical stimulant here. So from there onwards, from the retina, because it is full of nerve fibers, those nerve fibers together join together to form the optic nerve, which goes to the brain. So the optic nerve here, optic nerve. Now it carries these impulses to the brain. Now what happens at the retina? This retina acts as a screen. It is a screen-like action. It shows a screen-like action. So what happens here? The image is formed. What kind of image? Real image. Real image. Inverted image. Inverted. Now all this information via the optic nerve, it moves towards the brain. Which area of the brain? The occipital area of the brain, the vision area of the brain. Now in this area, the brain over here, it perceives these visual impulses, it perceives, it understands them, it analyzes them, it understands its shape, size, structure. There actually what we say the word seeing, actually seeing happens here. And the image here is what kind? It is erect. It's not like the retina inverted, it is erect here. So the actual seeing happens here in the brain after the brain analyzes these visual impulses coming via the optic nerve. In humans, the eyes are set in front of the face. We have two eyes. So each eye, it sees the object slightly different from the other eye. So the orientation of the object in each eye, the image of the object in each eye is slightly different from the other eye. Now what happens after this? Each eye forms its own image on its retina. The brain interprets these images as a single impression. We have binocular vision, also known as stereoscopic vision in humans. Now see, the ability to perceive distance and depth. Why is it? It is due to simultaneously focusing of an object in both the eyes. After that, the brain overlaps. Overlapping of the images takes place in the brain, and then this gives us the three-dimensional effect, which as we are looking at an object, we can perceive distance, depth of the object. Now sometimes some people complain of doubling of the image. Why does this happen? If the brain does not correlate the images of both the eyes properly, there is a problem there, then there is doubling. There is also doubling if the alignment of the eyes is not proper. Then also that person can complain of doubling. Another reason of doubling is when the visual center of the brain is inhibited because of alcohol. So these can be, so again if a question is asked to you, an alcoholic person sees double images. So you can say that the alcohol, it inhibits the activities of the visual center, hence there is doubling of images. Let us now study accommodation. When I say the word accommodation, it means the ability of the eye or the adjustment of the eye to assure clear vision of objects placed at different distances. So how does the eye achieve this accommodation? Two things can happen. Either the object can be close, it can be near vision, or it can be in distant vision. First let us study near vision in detail. Now see, for you I have drawn here the structure of the eye. Look at the object, it's close to the eye, near vision, close to the eye. The light rays are entering, here they are converging, there's more convergence here, passing through the pupil, this opening, and it's falling on the lens, and the image is being formed on the retina here. Now how is this achieved? When an object is placed at a close distance, at a near distance, how is near vision achieved? These are the parts of the eye that handle this process. The ciliary muscles, the suspensory ligaments, and the lens. Now I have drawn for you another diagram here. How does near vision happen? Now these are the ciliary muscles. Ciliary muscles. What happens to the ciliary muscles? These ciliary muscles, they contract. Hence they push the ciliary processes forward towards the lens. So remember, when you are comparing near vision and distant vision, remember that the ciliary muscles here will contract. Now what happens? When the ciliary muscles contract, this is the suspensory ligament. Suspensory ligament. These suspensory ligaments, they will relax. When they relax, the tension here on them is relaxed. So this is the eye lens, the lens of the eye. This becomes more convex. So it becomes thicker, it becomes convex. Again I am repeating, ciliary muscles contract, suspensory ligaments, because of the contraction of ciliary muscles and the ciliary processes push it forward. So towards the lens, what happens? The suspensory ligaments, they slacken, they relax, and the lens becomes more convex. Once the lens becomes more convex, the focal length, what happens to the focal length when the lens becomes more convex? The focal length decreases, thereby assuring near vision. So when you are learning near vision, please make a comparative table between the near vision and the far vision. What happens to the ciliary muscles? What happens to the suspensory ligaments? And what happens to the lens? Now let us look at how distant vision happens. In the diagram here, you can see the light rays coming from a distance. As they are entering the lens, entering the eye, they are converging, and the image is being formed here at the retina. How is this brought about? Again, the same parts of the eye are involved in ensuring that we have a clear distant vision. It's accommodating, so it has a clear distant vision. These parts are ensuring it. Now let us look at the diagram here. Ciliary muscles. What happened in near vision? the ciliary muscles contracted. But in distant vision, the ciliary muscles now relax. The suspensory ligaments, they will contract, pulling the lens. The lens is pulled. If you look at the distant vision, you can see the lens has become very thin. It becomes less convex. When it becomes less convex, its focal length increases, thereby ensuring a clear distant vision. Here is a comparative table for you. You can draw a same kind of table in your notebook. You can use your own kinds of words to understand, but this is basically to guide you on. Now let us look at the main structures of the eye that are involved in accommodation. Ciliary muscles, suspensory ligament and lens. Now let us see what happens when we are using the distant vision, the near vision or the distant vision. See in the near vision what's happening to the ciliary muscles. They are contracting. When they contract, they push the ciliary processes forward towards the lens. So what happens? The suspensory ligaments at that time, they relax. The lens, what happens to the lens? Because the suspensory ligaments have relaxed, the lens becomes thick, it becomes convex. Once it becomes thick and convex, the focal length of the lens decreases, thereby ensuring clear near vision. Now let's come to distant vision. When we compare it again here, ciliary muscles, what is happening here? Relaxed. The ciliary muscles are relaxed. The suspensory ligaments, they contract, they pull the lens and they make the lens thin, thereby reducing its convexity, and the focal length increases, thereby ensuring clear distant vision. So you can be asked questions based on criteria. What happens to the ciliary muscles? What happens to the suspensory ligament in each of these cases? What happens? What does the lens become like when we are using the near vision, when we are in distant vision? So please learn this as a comparative table. Let us study adaptation of the eye. To define adaptation of the eye, it is the ability of the eye to adapt to changes in light intensity. We can study this under two conditions, darkness and bright light. So let us see how the eye adapts in the dark and how it adapts when there is bright light. Now here I have listed three main parts of the eye, or pigments are contained in the retina. So let us see how this works. Pupils, when we are going to the dark, the pupils dilate. They open wide. Why do they open wide? Because they have to allow more light to enter the eye, so that to enable vision. Here, when we are in the bright light, the pupil will constrict. Because so much of light is already available, so to ensure that enough amount of light enters, the pupil will constrict. Now, what is the pigment? What is happening to the pigments of the eye? When a person is seeing things in bright light, we have the pigment visual purple. It's contained in the rods, visual purple, also known as rhodopsin. What happens to this visual purple pigment? It gets bleached. So it becomes less sensitive. So as it gets bleached. Now, if you are going to the dark, the visual purple or the rhodopsin, it gets regenerated. So I am writing VP for visual purple. What happens to it? It gets regenerated. Then the eyelids, when a person is in a dark place, naturally the eyelids will tend to open wide so that the pupils get more light and so on and so forth. Here in bright light, what happens as we go into from a dark place to a sunny place? The eyelids, they tend to close a bit so as to they can control the amount of light entering the eye. How do we see objects in their colors? That means how do we perceive color? The cells, these cones are present in the retina. Cones are responsible for our color vision. Now how this happens? Look at the features of the cones. Cones are stimulated by bright light. They are of three types, selectively responsive to different wavelengths. Now if we go out in moonlight, we cannot perceive colors. We cannot see different between yellow, red, green. Why? Because cones again are stimulated by bright light. The same objects, if they are looked at in day in bright light, we can easily see the difference in their colors. Hence, the cones which are found in the retina are responsible for our color vision.